Imagine your body for a second. It's basically a massive, bustling metropolis. And inside every single building of that city, you've got these microscopic factory workers just tirelessly assembling, breaking apart, and rearranging all the raw materials that literally keep you alive. Well, these workers are enzymes, and honestly, they are the unsung heroes of our biology. But here's the catch, and it's a big one. These micro-factory workers, they are absolute perfectionists. I mean, they require incredibly specific, perfectly tuned conditions just to survive, let alone thrive. So welcome to this explainer. Today we're going to dive deep into exactly what makes these biological machines tick, and we'll see what happens when their carefully balanced world gets disrupted.
Here is a quick look at our syllabus for today. First, the perfect working environment. Second, supply, demand, and concentration. Third, enzyme inhibitors. Fourth, enzyme kinetics. And finally, some really cool, real-world enzyme hacks. All right, let's jump right into Section 1, the perfect working environment.
Let's start by looking at just how ridiculously sensitive our microfactory workers are to their ambient surroundings. And the single most critical factor here? Temperature. At low temperatures, the molecules in our factory are just moving kind of sluggishly. Because they're moving so slowly, there's a really low frequency of successful collisions between the raw materials, which we call substrates, and the enzymes themselves. But as things heat up, the molecules gain kinetic energy. They start zipping around faster, colliding way more frequently, and with a lot more energy. So the rate of reaction climbs steadily right up until it hits a peak. That peak is what we call the optimum temperature, which for this particular enzyme sits right around 35 degrees Celsius.
But wow, look at what happens right after that peak. The reaction rate doesn't just gently slope back down, it literally plummets off a cliff. This sudden, dramatic drop is due to a process called denaturation. When the environment gets too hot, the structural bonds that hold the enzyme in its incredibly precise 3D shape, things like hydrogen bonds, they literally just start to break apart, and this permanently damages the enzyme's active site. If that active site loses its specific shape, well, the substrate can't bind to it anymore. The factory workers' tools are completely broken. For human enzymes, this total shutdown usually happens the second temperatures push past 40 degrees Celsius.
So temperature is obviously crucial for kinetic energy and keeping that structural integrity intact. But what happens to our microfactory if the pH isn't just right? Well, unlike temperature, a change in acidity doesn't necessarily slow down how fast those molecules are moving around. Instead, it directly messes with the ability of the substrate to actually bind with the enzyme in the first place.
The really crucial takeaway here is that an enzyme's optimal environment is directly tied to exactly where it lives and works in your body. Take pepsin, for example. Pepsin is an enzyme that works in the human stomach. And the stomach? It's an incredibly harsh, highly acidic environment, just full of gastric juice and hydrochloric acid. Because of this, pepsin has actually evolved to have an optimum pH of 2. It absolutely thrives in a highly acidic environment that would instantly denature pretty much any other enzyme from anywhere else in your body.
Moving on to Section 2, supply, demand, and concentration.
Okay, so let's say the temperature and the pH are absolutely perfect. Production still ultimately depends on the supply chain of our microfactory, right? We're talking about the raw materials, the substrates, and the workers themselves, the enzymes. When we hire more workers, the initial rate of reaction goes up in a perfectly straight linear line. As long as there is an unlimited supply of raw substrate available, adding more enzymes just introduces a greater number of active sites into the mix. And more active sites simply means a much greater likelihood of those enzyme-substrate complexes forming.
But let's be real, in the actual world, resources are rarely unlimited. So what happens when our microscopic workers run out of raw materials? Or on the flip side, what happens if we have a fixed number of workers, but we just keep dumping piles and piles of raw materials right onto the factory floor?
Well, when the amount of enzyme is fixed, adding more substrate initially causes a really steep rise in the reaction rate, but eventually the curve completely levels off. This plateau right here, this is the point of active site saturation. At this exact moment, every single enzyme worker in the factory is totally swamped. They're fully occupied and working as fast as they possibly can. Throwing more substrate at them at this point isn't going to speed anything up because those extra molecules literally have nowhere to bind. They just have to wait in line until an active site finally frees up.
Which brings us to Section 3, molecular saboteurs, better known as enzyme inhibitors.
Up until now, our microfactory has been running pretty smoothly, but biology is always full of conflict. Enter the molecular saboteurs, enzyme inhibitors.
These are molecules specifically designed to slow down or completely shut down our factory workers, and they basically use two main tactics. First, we have competitive inhibition. This is where the inhibitor has a physical shape that is super similar to the actual substrate. It essentially acts as an imposter, sneaking in and physically blocking the enzyme's active site so the real substrate can't even get in. Then we have non-competitive inhibition, which is honestly a bit more devious. This inhibitor binds to an entirely different part of the enzyme, an alternative site, but when it binds there, it forces the entire enzyme to change its shape, effectively warping the active site so it becomes completely useless to the real substrate. The difference between these two tactics is huge. With competitive inhibitors, because they're actively fighting for the exact same parking spot as the substrate, you can actually beat them just by flooding the factory with more substrate. Eventually, the sheer, overwhelming number of substrate molecules will outcompete the inhibitors, and the factory can still hit its maximum production rate. But non-competitive inhibitors, they are permanent saboteurs of that specific enzyme molecule. No matter how much substrate you add to the mix, those warped active sites just won't work anymore. So non-competitive inhibitors permanently lower the maximum possible rate of reaction.
Now, you might be sitting there thinking inhibitors are strictly bad news, right? But actually, they are absolutely essential for our survival. Reversible inhibitors act as crucial regulators in our metabolic pathways. Imagine a factory that's just churning out way too much of a product. In biology, the end product of a reaction can actually act as a non-competitive inhibitor for the very same enzyme that started the whole process. It binds to the enzyme, changes its shape, and temporarily just halts production. Then, as your body uses up that product, the inhibitor detaches, the enzyme returns to normal, and production kicks right back up. It's this incredibly elegant, automated feedback loop that stops our cells from blowing through precious energy and resources.
Let's look at Section 4, Measuring Efficiency Through Enzyme Kinetics.
So we know how the factory operates, and we definitely know what slows it down. But how do biochemists actually measure a specific enzyme's efficiency in a lab? Well, they use a mathematical model called Michaelis-Menten enzyme kinetics. This curve you're seeing represents the reaction velocity as substrate concentration increases. Notice that top dashed line labeled Vmax. Vmax is the absolute maximal rate of reaction when the enzyme is completely saturated with substrate. Basically, when every single active site is full. It is the ultimate speed limit of our microfactory.
But scientists also need to know how easily an enzyme actually reaches that top speed. To figure that out, they look at exactly half of the maximal rate, which is written as 1/2 Vmax. If you trace a line from 1/2 Vmax straight down to the horizontal axis, you hit a very specific substrate concentration called Km, or the Michaelis-Menten constant. Km simply represents the exact moment when exactly half of the factory's active sites are full.
So, here's the really crucial point to take away from this. There is an inverse relationship between Km and enzyme affinity. Think about it for a second. If an enzyme has a very low Km, it means it only takes a tiny amount of substrate to reach half of its maximum speed. This tells us the enzyme has a highly powerful attraction, a high affinity for its substrate. It's incredibly efficient, basically able to snatch up scarce raw materials with total ease. Conversely, a high Km means the enzyme requires a massive towering pile of substrate just to get going, which indicates a really low affinity.
Finally, Section 5, Real-World Enzyme Hacks.
Now that we've mapped out the intricate biology and the math behind these microfactories, it's time to see how humans have actually learned to hijack them for our own benefit. Out in nature, enzymes are mostly free-floating in liquid solutions, just randomly bumping into substrates. But for industrial manufacturing, having free-floating enzymes permanently mixed in your final product is super messy and really inefficient. So scientists developed immobilized enzymes. These are enzymes that have been physically locked in place, bound to an inert, stationary, and insoluble material, something like alginate. So instead of just mixing everything together in a giant vat, the raw substrate is simply passed right over the stationary enzymes, and the finished product just flows out the other side.
And the advantages of this little hack? They are massive. First off, there is zero enzyme left in the final product, which means it's totally uncontaminated and you don't have to pay for expensive filtering. Second, those immobilized enzymes can be reused over and over again. That's incredibly cost-effective because, let me tell you, biological enzymes are notoriously expensive to produce. And finally, and this is just brilliant, the very act of binding the enzyme to a stationary material actually makes its 3D structure more stable.
This means immobilized enzymes have a much greater tolerance for extreme changes in temperature and pH without denaturing.
A really brilliant, everyday example of how we use this technology is the production of lactose-free milk. Roughly 5 to 10% of the UK population is lactose intolerant, right? Their bodies just lack the specific enzyme needed to break down lactose, which is a complex sugar found in dairy. To solve this, the food industry simply takes the lactose-breaking enzyme, immobilizes it onto alginate beads, and then just flows regular milk right over it. The enzyme breaks the lactose down into simple, easily digestible sugars like glucose and galactose, leaving us with sweet, perfectly uncontaminated, lactose-free milk.
By understanding the perfect temperature, that delicate pH balance, the sneaky role of inhibitors, and the precise mathematical kinetics of these tiny workers, we've basically moved from simply observing biology to actively engineering it. Which leaves us with a pretty fascinating thought to end on. If we can already immobilize and hack enzymes to completely change the very food we eat, what other microscopic factories inside of us are just waiting to be engineered next? Thank you so much for exploring the microfactory with me in this explainer.